screen कर देना. Yes. So this is open nucleus breeding system. Now that is that you know that is a breeding system. It is the application of the principles of genetics and population genetics, in particular, along with the some statistical methods
Bioinformatics techniques
for increase in the for genetic improvement of the farm animals, or you can say genetic improvement of the farm animals, plants, uh, everything is known as breeding system. But when we are talking about the animal breeding system, then we have to tell that is for animals, because it may be the plant breeding system, it may be the animal breeding system, plant breeding, animal breeding, and even the poultry breeding also. But breeding system is a common term. When you just apply it in case of livestock, then we call it animal breeding system. When we apply it for poultry, we call it the poultry breeding system. And when we apply the same basic principles is same. When we apply the principles in case of plant, we call it the plant breeding system. So that nucleus breeding system, that you know that nucleus is the nucleus कहाँ रहता है? In the center of the cell, है ना? Center of the cell में क्या रहता है? Nucleus रहता है. So nucleus is, it is the everything is there, that is which can control the all function of the cell. ठीक है ना? So that is DNA is there, RNA is there, RNA is produced from the DNA. So and later on they shifts to the cytoplasm, but it is produced by the DNA of the nucleus. So ultimately it is under the control of the nucleus. So a breeding system where a nucleus of the animals is there, which is used for genetic improvement of the farm animals. So in the earlier case that we have studied, that is the conventional breeding system. That is conventional breeding system. The conventional breeding system is, it means select the best individuals, best male and best female should be selected among the lot, and they will be used to produce the progeny for the future generation. That is called conventional breeding system. ठीक है ना? Conventional breeding system, that is select best and mating best to best. That is called selective breeding. That is our conventional breeding system. And under conventional breeding system that we have read, that is a basic the basis of the selection or the criteria of selection. And one of the best criteria of selection is the progeny testing for improvement of the sex limited traits, particularly for the milk production. Clear? And with the help of this conventional breeding system, that is progeny testing, the lot of improvement in the milk production has taken place, not only in our country but across the world. But there is significant achievement or significant improvement is there in the developed countries, but a similar space of improvement is not taken place in the developing countries like India. So let us see what are the lacuna are there in our country, so that we cannot achieve that significant achievement in the genetic improvement of the farm animals with the help of this conventional breeding system. The reasons for non-significant contribution are, that is, first is non-availability of sires of high genetic merit. You know, that is for improvement of any breed, there is requirement of the male and female, but importance of the male is much more than the female, as I have said. That is, sire is called the half of the herd. One sire is sufficient to cover all the cows in a herd. If there, even if there are the 100 cows up there of a particular breed, so one sire is sufficient to cover all the 100 cows. So that's why that is the importance of the sire. So if sire is very bad, that is its genetic contribution is very less, then naturally the production level, the average production level will be very less, And if your sire is genetically very superior, then there is every possibility for increase in the productivity over the generation. So that's why that is the, so not only that, the suppose the good sire is available, but not the sufficient quantity, because in a particular breed, we want the improvement of a particular breed. There are, several number of breeds, uh, um, uh, up till now, there are 50 breeds of cattle in our country. So we want the improvement of all the 50 breeds. For that, we have to have the sufficient number of uh, sires of good genetic merit. ठीक है ना? Sufficient number of sires of good genetic merit भी होना चाहिए. Otherwise, if you have the very less number of sires of good genetic merit, then it will not be possible to cover all the cows of a particular breed by a very few number of sires. So that is that lacking is there in our country. That is non-availability of sires of high genetic merit in required number. Then poor spread of AI structure, AI infrastructure. So for speedy spread of the genetic improvement, the AI is the very important tool. That artificial insemination program. So one has to, there is a requirement of AI throughout the country, across the country. Uh, only in that case we can go for high speed of improvement. Very quick improvement can be achieved. Generally, we can see that is AI program is in most cases it is conducted in the urban, peri-urban region, and in the remote village it is very rare, because that is there is due to the lack of infrastructure for AI. What structure is required? That is not only that is straw or semen is not the sufficient. For that, the person is required, the trained person who will conduct the artificial insemination program, and that government machinery should be there. That where that artificial insemination center should be located at different places, in a village level, in a panchayat level, and only that in under that conditions that it can be done. And even a more important part is the trained manpower. If untrained manpower is there, he will not be able to achieve the success, because he does not know that where the cement is to be deposited. He does not know how does not know to how to judge the quality of the cement at the time of the insemination. So how to thawing the straw when it is taken up from the fridge, or from the liquid nitrogen. Then so all these things, so some knowledge is required. That's why trained manpower is essential for uh, for um, uh, for successful artificial insemination program. That is lacking is there in our country. Then small size of farmers herd. For any breed, say for example the Haryana cattle breed. So the Haryana breed, that is home track is Haryana state, but Haryana cows are available in most of the state of the country, and but the number of animals of that particular breed is very large. Very large number of animals are there, but the farmers generally keep one animal, two animal, three animal, four animal, like this. That means farmers herd size is very small. Wherever in case of the developed country, you will find that even a farmer is maintaining that 400 500 animals of a particular breed. So that they are the farmer. But in our case, that farmers are very poor, so they are maintaining only 1 2 3 4 5 animals, not more than that. Mostly 1 to 2 animals. So that is the herd size of farmers. Herd size is very small. That is also the hindrance. Not only that, farmers herd size, even the herd size in the organized farm, that is also very small. Organized farm means that is the institutional farm, that is farm maintained by the government. that is government cattle farm is there, government buffalo farm is there. That is everything is the management is taken place by the government itself. That there is a lot of land is there for cultivation of the fodder, but even then, in most of the cases, that fodder land is not sufficient to produce the sufficient quantity of fodder to feed the animals. That's why these are the some lacuna. High cost of data recording when farmers are maintaining the animals, so that our farmers are not so educated, so their lack of sense is there, so they are not able to maintain record the data. That data means that is the age at sexual maturity data is to be recorded when the animals come and heat. So that is the when the animals are inseminated, the date of insemination, when the animal is expected to calve, that is date of calving, and that gestation period, that's calving interval, all this data to maintain of the animals. So our farmers are not able to maintain the data. So for data recording, you should have to engage some enumerators for data recording. So you have to for a particular for improvement of a particular breed, you have to engage a large number of enumerators. That required that is costly affairs. So that is a high cost of data recording. Even the data recording may not be very authentic under the farmers herd level. And then the selling or keeping of animals, it is totally depends upon the mentality of the farmers. If the animals are not well, or the animals are not giving meals in sufficient quantity, or the animals are not coming in heat in the right time, or animals are not giving birth a calf every year, so the farmers may sell the animals so before completion of the data recording. So these are the some problems or the lacuna in the in the village or the farmers herd with the farmers. That's why we cannot able to organize the breeding improvement program. So this is about the structure or the feature in the farmers at the farmers level. Now, the animals are also maintained by the government under that is called organized herds. So, what is the lacuna in the organized herd? Organized herd is also very small in size, the small population size, and the less intense selection. So, due to small population size, the selection intensity cannot be increased. And until or unless you increase the selection intensity, then the improvement cannot be achieved as per the level of desires. Then putting emphasis on non-productive traits. So sometimes for the selection of animals, some non-productive traits are given emphasis. So these are the reasons. That's why we cannot, we could not be achieved in the genetic improvement of the farm animals through the breeding system, that is prevailing breeding system, which we call the um, conventional breeding system. Now, for that, some new concept has come up, which is known as nucleus breeding system. This now let us see what is the nucleus breeding system. So a nucleus that is a farm is there in the institute, the government, a farm is there. When we conduct the poison testing program, the entire farm or entire herd is considered as the one. Okay, but when we are considered for the nucleus herd, when the concept of nucleus herd is there, that is the entire farm is there, we have a pocket, or we can call it the nucleus of the farm, that it will maintain only very few number of elite animals. That is suppose if we consider for the milk production, so the milk production will be the criteria, those females giving the best high quantity of large quantity of milk, they will be kept in the nucleus. And this is the concept of nucleus herd. Only 10 to 15% of the total animals strength of the farm will be uh, will be considered for the nucleus, and that is called nucleus herd. So nucleus herd is created, which is used for production of superior males for breeding purpose in the population. So that all the elite animals are there in the nucleus herd, elite females. That means the top ranking females of the herd are present, are kept in the nucleus, and only what is the percentage? That is 10 to 15%. If there are 100 cows are there in the farm, so only 10 to 15 cows will be allotted for this particular breeding program, that is nucleus breeding system. And whatever the male cup will be produced from these 15% cows, they will be maintained and they will be raised to be the bull for reproduction purpose. So when a breed of livestock is kept in the institutional farm, it is known as organized farm or organized herd. So when animals are of a particular breed are kept by the farmers, then we call it the commercial herd. The commercial herd, it's got different names are there. Sometimes we call it commercial herd or commercial flock. कभी हम लोग बोलते हैं कि village herd or village flock, or sometimes we call it peripheral herd. Why we call it peripheral herd, I will explain later on. So now you know that the animals maintained by the farmers, that is that the 1, 2, 3 animals are maintained by the farmers. There's very few number, so they will be called the commercial herd or the village herd. So an organized herd under position testing program is treated as the single herd, but under the nucleus breeding system, we divide this total herd into the two groups. One is called the nucleus herd. The rest is called the test herd or the multiplier. Test herd or multiplier. The nucleus herd is constituted with the elite females. So this is the diagram. You see the schematic diagram of nucleus breeding system. So this is this is the farm or the organized farm. If this organized farm is divided into the two. One is the centrally you see the nucleus herd, which is constituted with the 10 to 15% elite females of the total herd strength, and the outside the periphery of the nucleus you see this is the called test herd or the multiplier herd. That means that whatever the genetic improvement is there in the nucleus herd, that means the best superior sires are produced from this nucleus herd. They will be allowed for the breeding purpose. So where they will only here are only very few females are there, only 10 to 15% female is there. So if they produce a calf, only 10 to 15 calf will be produced per year, because if there are 100 cows in the total in the herd strength is 100, so out of that only 10 to 15 cows will be there in the nucleus herd. That means it is expected that 10 to 15 calf will be produced, even if we consider the sex ratio. So out of 15 means 7 or 8 female calves expected to produce, and 7 to 8 male calf is expected to produce. So those male calves will be sire for future breeding program, and these sires will be used for breeding purpose in the this in the with the rest of the animals of the farm. This in the rest of suppose that if out of 100, some 15 cows is there in the nucleus, and rest 85 cows are there in the test in the outside the nucleus. So the animals present outside the nucleus, they are called test herd. That means यहाँ पे जो भी आपका सायर पैदा हुआ है, they will be tested not only for testing but also for multiplying. So all these 85 cows are there. They will be inseminated with the inseminated, and then they will produce the calf, and so many calves will be produced, so many sires will be produced for future use, and those sires will be produced from this multiplier herd or test herd. They will be used for genetic improvement in the to the animals kept by the farmers, and that is called the village herd. Sometimes you call village herd, sometimes you call the peripheral herd, sometimes you call the peripheral commercial herd. Since the village herd are the related with the profit, so therefore it is called the commercial herd, And since outside the farm or the farm, the whatever the farmers are there, that's why it is called the peripheral herd. ठीक है ना? So the village herd are called also the called commercial herd, village herd, or are also called the peripheral herd. But whatever the genetic improvement will be there in the nucleus herd, they will be transferred to the this test herd or the multiplier herd, where the animals will be multiplied, because these sires, the sires will be produced in the nucleus herd generally will not be transferred to the village herd. Only they will be allowed for breeding in the test herd for the production of the male calf, which will be used for breeding purpose in the at the village level. This is the concept of the nucleus breeding system. So the nucleus herd, in nucleus herd, the breeding of male and female takes place within the herd itself. Now, whatever the genetic gain achieved in the nucleus herd is passed on to the test herd or the multiplier herd, and from multiplier herd to the village herd. That is, जो भी improvement will be there in the nucleus herd, that improvement will go on to the test herd or the multiplier herd, and from multiplier herd to the village herd. This is the concept. And now, depending upon the flow of genes, the nucleus herd is of two types. One is closed nucleus breeding system, other is the open nucleus breeding system. So what is closed nucleus breeding system and what is open nucleus breeding system? You see, in case of closed nucleus breeding system, the flow of genes is unidirectional. Flow of genes is unidirectional. That means the genes is flows from the nucleus herd to the test herd or the multiplier herd, and from the multiplier herd to the village herd. That means male calf will be used, or the sire will be used for breeding in the multiplier herd, and whatever the sires are produced from the test herd or the multiplier herd, they will be ultimately used for breeding in the village herd. So that means that the genes is being flowed from the top to bottom, that is nucleus herd to the peripheral herd, from peripheral herd to the village herd, and that flow of genes takes place in one direction. That is called unidirectional, and that's and the genes, that means the superior animals sometimes present in case of the with the farmers, so they will not be allowed to come in the nucleus herd. That's why it is called closed. So that means it is the one-way traffic. This is one-way traffic, not by-way traffic. Okay, na? So this is a nucleus breeding system. And what is open nucleus breeding system? Under open nucleus breeding system, that means the nucleus is open. Why it is called open? Because the one way, that is the flow of genes takes place in bidirectional. That means initially, that is the animals, the superior sires are used for multiplication in the test herd or the multiplier herd. The calves will be produced, they will be used for breeding in the village herd. So this is the one way of traffic. And in the another way, the sometimes village are also maintain good quality of animals. So that the animals are also being transferred from the village herd to the seedhe nucleus herd. Nucleus herd, after of course the quarantine should be maintained. The animals will be kept in the quarantine for a few days to see whether the animals are suffering from any diseases or not. And then if the animals are not suffering from diseases, then all the good quality animals are to be allowed to enter into the nucleus farm. So that means the genes are flowing in both the ways. That means here nucleus is open, not closed. That's why it is called open nucleus breeding system. Clear? There are two nucleus breeding system. One is closed nucleus breeding system and open nucleus breeding system. Under closed nucleus breeding systems, the gene is flows from the top to the bottom. That is from nucleus herd to the test herd to the village herd. Whereas in case of the open nucleus breeding system, the nucleus is open. It means the genes are being flowed from the nucleus herd to the multiplier herd to the village herd, and the reverse is also taken place. That is village to the विलेज टू द डायरेक्ट न्यूक्लियस हर्ड। विलेज से न्यूक्लियस हर्ड आता है, विलेज से मल्टीप्लायर में नहीं आता है, न्यूक्लियस हर्ड भी लिया जाता है आफ्टर टेकिंग द क्वारंटाइन पीरियड। So the whatever the genetic gain takes place in the nucleus herd, that is passed on to the test or multiplier herd, and from multiplier herd to the village herd, and then as a result, there will be the overall improvement of the breed in the state or in the country. So this closed nucleus breeding system, that is already told, that is unidirectional. Then whatever the genetic improvement taken place in the nucleus herd, that will be transferred to the test herd, and from the test herd to the village herd. And gene never flows back from the village herd to the nucleus herd to avoid the risk of introducing disease in the nucleus herd. So nucleus herd में अगर कोई disease हो जाता है तो क्या हो जाएगा, आपका पूरा nucleus फार्म को आपको डिस्ट्रॉय करना पड़ेगा, पूरा आपका ब्रीडिंग प्रोग्राम खत्म हो जाएगा. Then you have to start fresh. That will take time. That's why generally this closed nucleus breeding system is used in case of the pig and poultry. Pig and poultry में use किया जाता है closed nucleus breeding system, and you know that the pig or the poultry is very sensitive. If the salmonella infection is there in the poultry or taken from the outside, naturally that entire flock will be affected with the salmonellosis. Or if there is the fowl cholera, is the you without testing the birds, or you are entered into the nucleus herd, so all the birds will die due to the fowl cholera or due to Rannichet disease like this. That's why the in case of closed nucleus breeding system, the animals outside the farm are not allowed, are not getting entry into the farm. Similarly, in case of pigs also. So this open nucleus breeding system, that is flow of genes, it is takes place bidirectional. So that is genes flows from nucleus herd to the multiplier herd, from nucleus herd to the multiplier herd, and from multiplier herd to the village herd, and vice versa. The superior animals available in commercial herd are also introduced in the nucleus herd, and इसमें एक फायदा है, क्या है, to reduce the rate of inbreeding. Suppose nucleus herd में very few number of animals are there, there is every possibility of inbreeding. So to reduce the inbreeding, but to avoid the inbreeding, we may allow the entry of the superior animals present with the farmers into the न्यूक्लियस हर्ड. वहां से न्यूक्लियस फार्मर से परचेज कर लेते हैं, जैसे हम लोग अभी अपना यूनिवर्सिटी के फार्म में यही करते हैं. कभी कभी हम लोग क्या करते हैं, विलेज में जाते हैं जिसके फार्मर्स के पास बहुत अच्छी गाय रहती है, जो मान लीजिए डेली 15 लीटर 12 लीटर 14 लीटर 18 लीटर दूध देती है, उसको लेकर आ जाते हैं इन द फार्म. फिर हम एवरी ईयर बाय एवरी 2 इयर्स इंटरवल जो हमारा एनिमल्स को ऑक्शन भी करते हैं. दिस इज अ सिस्टम इन द न्यूक्लियस हर्ड. सिमिलरली द विलेज हर्ड में फार्मर्स भी सेल कर देते हैं. अगर इफ द कीपिंग ऑफ एनिमल्स इज नॉट प्रॉफिटेबल, द फार्मर्स कैन सेल इट एट एनी टाइम. दैट इज लिबर्टी इज देयर, बिकॉज फार्मर्स खुद अपने एनिमल्स का मालिक है, वो कुछ कर सकते हैं. ठीक है ना. तो नाउ टू इंक्रीज द जेनेटिक गेन, another is to increase the genetic gain, because superior animals are also maintained by the farmers. So ONBS is mostly used in cattle, buffalo, sheep and goat. ठीक है. CNBS is used for sheep, pig and poultry, and ONBS is used in cattle, buffalo, sheep and goat. So ONBS can run in an organized herd at institutional level or by forming the breed societies. Even अगर village में अगर करना है तो